Hey guys, today we're going to be talking about the notochord and neural tube development of the early vertebrate embryos. Our first image we see here is going to be the blastula. This blastula is composed of the blastocoel, which is the opening we see in the middle, as well as blastomere cells, which are going to be represented with these blue balls. The blastomere cells will later develop into the endoderm, which will later in the embryo develop into the epidermis. After the bastula formation, the bastula is going to invaginate itself and gives rise to the archenteron. The archenteron is thought as a primitive gut, which will later as well develop to the actual gut. Here in this image, we can see the presumptive notochord cells starting to develop, are invaginating of the blastocoel, which makes it a bit smaller than before, as well as the archenteron opening and its respective cell line. After the invagination of the blastula, the early gastrulation process starts to begin, where the three different germ layers are going to differentiate into mesoderm, endoderm, and ectoderm. For our purposes, the ones we are most involved with are going to be the ectoderm, which used to be our blastomere cells here in blue, which will now develop into the outer surface cells as is the epidermis. And we are interested in our notochord development, which will be the mesoderm. We can see from this image that the blastocoel opening has now disappeared, and our biggest opening is going to be the archenteron. As well as this opening, you can see that the dorsal and ventral lip of the blastopore is starting to appear. The blastopore is going to be an opening between the outside of the embryo and the inside that will later in gastrulation almost ultimately close. In late gastrulation, the three germ layers have developed further. In this image here, we can see how the ectoderm, which is represented with our blue balls, is almost fully around the embryo with the exception of the blastopore that leaves an opening for communication between the inside and the outside of the embryo. In orange here, we can see the endoderm cells as well as their archenteron opening in the middle, as well as the future notochord here represented in green. In the later stages of gastrulation, our three germ layers have almost fully or are starting to develop. Here we can see that our blue balls, which represent our epidermis, are almost all surrounding the surface of our embryo. The endoderm cells are inside of the embryo where they'll be located for the rest of development. And developing mesodermal cells are starting to appear on either side of the notochord. The notochord itself is a signaling tissue that helps the developing of the neural plate, which here is represented with their pink balls. The neural plate is a precursor to the central nervous system. After the plate, it will develop into the neural tube. Our three germ layers have fully developed. We can see here that the epidermis has surrounded the embryo, and the neural plate and the notochord have been dragged inside and are starting to develop by mitosis. The mesoderm is also developing, and it's going to later turn into the somite cells, which is a mesodermal segment that will help the neural tube development. This orange middle, the hollow orange middle, is here going to be called the archenteron, and it's going to be part of the gut with the help of the endodermal orange cells, as we can see here. After pouch formation, the somite formation starts to occur from the mesoderms. We can see here two somites represented with these purple circles, and their openings called the coelom. These somites are going to help the neural plate develop in the neural tube, as well as the notochord and the endoderm with their migration in order to create the gut and the fully developed notochord. Lastly, our narration process begins. Here you can see that the neural tube has fully developed. It's going to be this pink structure with this opening that will later develop into presumptive neural tissues of the central nervous system, like is the brain and the spinal cord. The notochord here in green has fully developed, and it's going to be one of the key organ tissues in this embryo that will help all developing tissues differentiate into the determined cells that they need to be. The gut here has fully developed from the endoderm, and we can see it in this circular orange structure. Our mesosome has as well developed and encloses the inside of all the tissues of the embryo.